Okay. All right. Let's start our second part of the workshop today. Right now, Kevin's gonna give you us a tour on the biology laboratories. And Kevin, if you want to share our screen.
Can you see it okay?
Yes.
So as Luke mentioned earlier, you can get to these tours from the main Neutron Sciences page. The labs are a little bit difficult to get to, but if you start the tour at the SNS, once it zooms in here, if you go around the corner, there's these orange dots and you can pick out the labs. So I'm going to kind of blow through this fairly fast because we're kind of behind schedule, but you can come back later and look around at all the user labs here at the SNS. But I'll start here outside of the biology laboratories. The main lab is the biodeuteration laboratory. And by the way, all these biology labs are a shared space that support all of the biological neutron scattering program here at ORNL. And the biodeuteration lab has capabilities for protein expression purification with a special focus on biodeuteration using batch and fed batch cultivation of E. coli and yeast, because those are the two main organisms that we use for producing these biomolecules. So in general, it's a basic biochemistry lab with a few extras that focus on being able to produce multi-milligram scale quantities of material for neutron scattering experiments. And if I stop by the bioreactor area, we have two different systems. One is a small scale parallel bioreactor system on the left, can do 4 vessels from around 60 mils to 250 mils. But the workhorse is the system here shown in the center, which can grow cultures from a low level of 750 mils up to 5.5 liter scale, depending on the vessel that we use. Move to the other side of the lab. We have various ACTA chromatography systems for purifying materials. And just like most basic structural biology labs, we have shaking incubators, preparative centrifuge for spinning down cultures. And then we have a cold room where we can do dialysis or maybe gravity-fed columns, sample prep. And then I'll move across to the other room next door. We use either sonication or high-pressure homogenization to break cells in this space. This Aviston homogenizer can flows at about 50 mils a minute. So you can put the cells up top and then flow them through, and that pressure drop as you pass through a small hole in the homogenizer allows the cells to be broken that way. So that's useful when we're doing the bioreactor runs if we produce a large amount of cell paste that can't be handled without multiple runs of sonication. I'm going to move across the hall on the way to the crystallization and characterization lab. We have this protein X-ray lab with the X-ray diffractometer on the right and the BioSax instrument on the left. And in this space, researchers can use either instrument to characterize samples either before or after their neutron beam time. And I'll pass over to the third lab in the biology lab suite, and that's the crystallization and characterization lab. So as I mentioned earlier, these labs support all the Neutron Biology program. This particular lab has several incubators for the crystallographers to grow cells or grow crystals for their experiments. Also has means for setting up those trays either manually or pipetting robots. We have a preparative centrifuge here for additional protein purification separations. And then being a characterization lab, we also have the ability to use absorbance fluorescence plate reader. This far side of the lab, we have a CD instrument, UV vis. We use this laboratory to characterize our proteins using protein electrophoresis, and then we have gel imaging capability as well. And just like HIFER has an area for doing biohazardous work, we have a BSL-2 capable lab here as well at the SNS. But we also use this small space for insect cell expression to keep this area separate from what's going on in the other lab with E. coli so that we're not cross contaminating between the two cell types. So that's a brief overview. Hopefully that gets us close back on schedule. Wellington?
Yes. Thanks so much, Kevin.